the roofs. The Fasano twins' grandmother actually was a shortwave radio operator, and she had a really extensive antenna that came off the roof that had a much bigger footprint, and that was used for shortwave radio. And because we used to play football in the street, we would steal roadblocks, wooden roadblocks, and wall off the street. And so they always called the cops, and they put a policeman would show up and stand in the intersection and drive us away. Now here's a little bit of a lie. He wore a magnesium badge. And he kept an eye on everybody. So if you take a look at this molecule of chlorophyll, and you start in the upper left-hand corner, and these quadrants are actually numbered. Each one of these nitrogen-containing houses are called pyrrole rings. And so this structure is a tetrapyrrole. And if you were to go into chlorophyll and somehow magically remove all of the magnesium and replace it with iron, it would turn red. And if you look at the structure of hemoglobin, it is 4 proteins. Each protein binds a tetrapyrrole complex with an iron in the center rather than a magnesium in the center. So these molecules are related. So it's a tetrapyrrole. It has a lipid tail. And if you go around the ring, you get a double bond, a single bond, 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 and and it ends up being, oh, I did miss it. A resonance hybrid. So that when light strikes it, the electrons, being as part of a resonance hybrid, are freer to move, and this area can become excited and be able to transfer electrons. Now, for those of you who had Bob for plant biology, you'll remember that there are different kinds of plants in the world. There are also algae, and they are can be grouped based on the kinds of photosynthetic pigments they have and the various kinds of chlorophyll. There's chlorophyll A, B, C, D, etc. And the different chlorophylls are different because of the TV antennas on the houses. So these four at the top are the ones that are most variable. And in fact, you can distinguish all of the A, B, C's, and D's by R group 1, R group 2, and R group 3. So you're not going to have to draw this for an exam, but I did want to show you the resonance hybrid nature and how the different chlorophylls do differ from one another. And that chlorophyll A is the most important. It seems to be the chlorophyll that is, in quotes, the most ancient molecule, because all God's children that have photosynthesis have got to have chlorophyll A. The other ones are accessory pigments, essentially. And so when light strikes, there's excitation of the molecule, and there are a wide variety of other pigments. And another sketch although I may not have the right number of peaks. But this is looks similar to carotenoids, carotene, and they too have essentially extensive regions of resonance, and the carotenoids are lipids that fall into that simple lipid class, where 1, 2, 3, 4, 5. This is an isoprene unit, 5-carbon compound, and essentially lots of isoprene units are linked together in really different structures that give rise to the carotenoids. If you look at a photosynthetic membrane, I probably have too many peaks here. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah. So there's supposed to be 1, 2, 3, 4, 5, 6, 7, 8. No, that's right. Hard to believe. If you break this in half, which would be here, this would be vitamin A, and this would be vitamin A. So carotenoid, carrots. It's a orange-yellow pigment which absorbs strongly in the blue and ultraviolet. If you think of this as a chain of carbon and start from one of the ends, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, or if you think of that as the chain, it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, which puts the carotenoid right there. Right here. In many books, you will see carotenoids and xanthophylls and phaeophytin, well, not phaeophytin, xanthophylls and other pigments that are associated with photosynthesis you'll see them referred to as accessory pigments, and that they contribute to photosynthesis by broadening the range of light absorption. Not so true. What carotenoids and many of the accessory pigments do is they absorb light that is not absorbed by chloroplasts, but they don't transfer that excitation over to the chlorophylls. Instead, essentially what is happening is that they act as sunblock because leaves have to be positioned so that they get the maximum exposure to light. Then they, like you, are sensitive to ultraviolet radiation damage. You don't stay out in the sun and constantly do this as the sun passes in the sky and expect to survive very long. And so the carotenoids act as essentially sunscreen for the chlorophylls, which would be in the membrane with their own lipid tails hanging down. If we look in detail, at a portion of a thylakoid, this being the thylakoid lumen, we're going to see a lot of similarities. I'm going to stress those similarities to oxidative phosphorylation. There are going to be light harvesting complexes. The light harvesting complexes are scattered across the surface of each thylakoid. And so if this is a portion of a thylakoid, here might be a light harvesting complex. That particular one is in a stromal lamella. And that light harvesting complex, if seen from above, look like this. With two chlorophyll A molecules in what is referred to as a reaction center that is associated with proteins. Associated with that reaction center these form what is referred to as the special pair. And regardless of where light strikes the light harvesting complex, resonance induction will cause the excitation to be transferred ultimately to the special pair. In the thylakoid lumen, there is a protein that contains manganese, Mn, as cofactors. And essentially what's going to happen is that light is going to excite the light harvesting complex constantly, photons constantly being bombarded. That is transferred to the special pair, and the special pairs